Hello.
Hey, hi, how are you?
How are you?
I'm good.
Good. Hello, Mehul.
Hi, Bradley.
Hi.
Hi, Soham.
Great. Hello, Morgan. Oh. Another Susan.
Hello.
Hi. Any updates from Susan or?
No, I'm supposed to be writing a paper.
Oh, yeah. Well.
Yeah. So I have to get going and do that. Other than that, no. Just yeah, it's supposed to be sort of a review paper.
Oh, really? For a journal or?
Yeah. On tissue and tensegrity for some reason. Yeah.
That's good. Yeah. Well, welcome. Yeah, last week, thank you to Mehul for going over his hypergraphs work that he's been doing, and he's been doing this with other people. And I like the direction it's going. Hopefully, we can continue down that road this summer. And of course, we have the DevoGraph repository for people who are interested in Google Summer of Code and maybe working on some parts of that Mehul talked about last week. Okay, so we had a comment from Soham. This is Morgan had sent me this paper which shows how a hypergraph extension makes a graph neural network better than a specialized topological model.
Yeah, so I was wondering if we could do a similar thing for the developmental graph neural networks which we have, if we could extend it to hypergraphs.
Maybe, yeah. Could you put a link to the actual paper in the chat? Is it an arXiv paper?
Yeah, I'm doing that.
Yeah. All right, so this is the arXiv paper that Soham and Morgan are talking about. This is enhancing the utility of higher order information and relational learning. And so let's go through the abstract and see what they're talking about. Looks like they're talking about multi-way networks. The abstract reads, higher order information is crucial for relational learning in many domains where relationships extend beyond pairwise interactions. So yeah, we typically think of our standard graph, our graph topology as a set of ith-jth connections. There are these pairwise interactions, and of course you can characterize things beyond pairwise interactions by looking at, say, like their neighborhood or their community or some sort of higher order construct. And we were talking about using hypernodes for that in hypergraphs. So this is, of course, what they're getting into with hypergraphs. Hypergraphs provide a natural framework for modeling such relationships, which has motivated recent extensions of graph neural network architecture to hypergraphs. However, comparisons between hypergraph architectures and standard graph level models remain limited. In this work, we systematically evaluate a selection of hypergraph level and graph level architectures to determine their effectiveness in leveraging higher order information in relational learning. So they're picking hypergraph level and graph level architecture. So I assume they're doing what they're doing with graph level is they're either comparing them or they're looking at different attributes of their connectivity. Our results show that graph level architectures applied to hypergraph expansions often outperform hypergraph level ones. So this is where they have like a standard graph level architecture and then a hypergraph expansion. I don't know how they're going to do that, but they talk about that in the paper. So this often outperforms hypergraph level ones, even on inputs that are naturally parameterized as hypergraphs. As an alternate approach for leveraging higher order information, we propose hypergraph level encodings based on classical hypergraph characteristics. So hypergraph level encodings, I guess, being the things at the level of hypergraph. So I think we've done this a little bit with the developmental hypergraphs where you have maybe your nodes are cells, and your hypernodes might be tissues or different regions. Sometimes they could be, like we talked about the last few weeks, we've been talking about lineage trees having sublineages as those hypernodes. And so, we can have these hypergraph level encodings. Usually people kind of do that in hypergraphs, but yeah, this is interesting how they're kind of approaching this. While these encodings do not significantly improve hypergraph architectures, they yield substantial performance gains when combined with graph-level models. Our theoretical analysis shows that the hypergraph level encodings provably increase the representational power of message-passing graph neural networks beyond that of their graph-level counterparts. So message-passing graph neural networks, this is where you have a graph and you're using a message-passing protocol, and there's standard methodologies for that. Basically, you're measuring not just the connectivity, but the messages passed between them, which could be like in a neural network, it's information being processed by the graph, but sometimes it's in where there's some communication in the graph, or where there's activation in the graph, so from one node to another. So you have these message passing protocols that are sort of designed for this, and it's not necessarily, I don't know how suitable it is for biology. Certainly people use message passing protocols for things like proteomics and other domains of computational biology. But it's interesting to think about. I don't know what the state of the art here is in message passing in biological applications, but it would be interesting to think about. So they actually talk about multi-way networks, and they bring this up. And this is something that kind of is an interesting topic. It's kind of a niche topic. So, this is where they're talking about using graphs to represent higher order information. And multi-way networks arise in many domains in the social and natural sciences, where downstream tasks depend on relationships between groups of entities rather than the pairwise relationships captured in standard networks. So they're actually, where we have this sort of nested set of information. So if you have, like, for example, a bunch of cells, they have multiple attributes, and you can you can build a network based on one set of attributes, but you also have these other alternate sets of attributes that you can then also organize these relationships by. Another thing is, like, when you have a cell, a developmental cell that has a certain role in development, and then it differentiates into some sort of an adult cell. You can capture those relationships with multi-way networks. They cite Bick 23, Benson 21, and Schaub 21. I'm not sure what those citations are. Well, what are higher order networks from Siam Review? Is the Bick from 23, and Benson 21 is higher order network analysis takes off, fueled by classical ideas and new data that's on the arXiv. And Schaub 21 is signal processing on higher order networks living on the edge and beyond. That's from signal processing. So there's some interesting papers here. A lot of interesting network theory papers in here as well, I see. So anyways, this enhanced flexibility has motivated a growing body of literature on extending classical graph neural network architectures to hypergraphs, including message passing and transformer-based models. So there's message passing and transformer-based models. Message passing is this UniGNN, a unified framework for graph and hypergraph neural networks. And transformer-based models is LEU 24, which is this hypergraph transformer for semi-supervised classification. So we're talking largely about we're kind of segueing to graph neural networks. Typical validation studies compare hypergraph architectures against each other, but not against standard graph neural networks. Here we perform a comparison of selection of both types of architectures. We observe that graph-level architectures strictly outperform current hyper-level ones, even if the input data is naturally parameterized as a hypergraph. In that case, the graph neural network is applied to the hypergraph's clique expansion, a natural reparameterization. Okay, so that's where we have, I guess, I don't know what, I'm not familiar with the term clique expansion, but basically we're reparameterizing the graph. So, and then so they raised this question: how can higher order relational information be effectively utilized for learning? So we have all sorts of information in these graphs, and the question is, basically we have this not just a set of relations, but this relational set of relations that makes any sense, where each node has like different attributes, and you can have these alternate topologies emerge. So let me see if they have, so they have a bunch of things in architectures here. They kind of describe them, overview of architectures. We have some math about hypergraph level architectures here. Transformer-based graph neural networks, message passing graph neural networks. And then they talked about these encodings that they're using. They're using structural and positional encodings. So this is where you have structural information and positional information. And both of these would be relevant to developmental biology, by the way. So the structural information would be, I guess, it could be nomenclature, it could be tissue type, it could be other things. Whereas positional would be something about like its position along an anatomical axis or its coordinates in space. So we have these different encodings, and we can then sort of use that as a way forward in here. Encodings can capture either local or global properties of the input graph. So you're actually taking an encoding of the connectivity matrix, if I'm understanding this correctly. And it depends on the sort of the set of relations you're interested in. I don't know if you have multiple encodings or not, but I like this idea of having an encoding on top of the connectivity graph. Empirical evidence demonstrates that incorporating these positional and structural encoding significantly improve the performance of GNNs. Okay, so you actually can use there are different types of approaches like graph Laplacians and other types of random walk methods that can be of help here. Then, of course, I talk about representational power or expressivity, which functions can and cannot be learned by the model. So this is, of course, we want to keep that in mind. So I guess this is an example here. So this is figure 1, a pair of graphs from the Breck basic category. The graph's liftings, the hyperedge sizes, and node degrees. So these are like, I guess, so your top left is your basic category. Your graph liftings are the top right. Hyperedge sizes are bottom left. And node degrees are bottom right. So you have count versus hyperedge size for the first two, node degree for the second two on the bottom. At the top, we have two graphs, graph A and graph B, and then on the upper right, hypergraph A and hypergraph B. So for the graphs versus the hypergraphs, we look at hyperedge A or hypergraph A edge degree distribution. So this is 13 hyperedges in this, and we have, or actually, wait a minute, this is graph hyperedge. I don't know why they're doing it like this. I guess they're taking hypergraphs from graphs here and graphs from hypergraphs here. But anyways, they show these for these different, I guess they're similar topologies, and they're showing the different hypergraph hyperage distributions, and then for this you have these hypergraphs, or these groupings, and then you have, so you have the hypergraph edge and node degrees here. You have the graph, the graph representations up at the top, and the hypergraph ones up here. So they all result in these different sizes and degrees. Okay. So that's kind of, I didn't really understand it, I guess, without really diving into the paper. But that's kind of one of the comparisons they're making here. This actually is, let's see. Okay. So they did a set of experiments, and then their discussion. Let's get to the discussion so we can think about this a little bit more. In this study, we investigated the performance of hypergraph level architectures in comparison with graph level architectures for multi-way relational learning tasks. Additionally, we proposed hypergraph level encodings as an alternate approach to leveraging higher order relational information. So that's what we're interested in. There's some lessons for model design, some limitations. So, okay, when they say lifted, they mean reparameterized. So a key limitation of the study is the lack of a benchmark consisting of true hypergraph structured data. Many of the existing datasets consist of graphs that are reparameterized or lifted to hypergraphs. So these are where you have graphs, like if we had a graph of the DevoWorm group, and we had connections between people in the DevoWorm group, we could then lift those data and create it. And this is kind of what we're doing with the developmental hypergraphs. We're lifting that, reparameterizing that to hypergraphs. So, that's that. But then I guess what they're suggesting is that you want to have a true benchmark that is an actual hypergraph to ensure that that lifting process, that reparameterization is on target. This suggests the establishment of better benchmarks is a key direction for future work. And they're interested largely here in topological deep learning, or at least, and that's something that we've talked about as well. That's kind of a goal for a lot of these approaches, is to get like these topological representations and topological data analysis and all this. We envision future benchmarks that are based on scientific data, such as these citations here, where multi-way interactions that are naturally parameterized as hypergraphs are known to arise. Another limitation of the study arises in the choice of hypergraph architectures. While our selection was guided by top-performing models and reached in benchmarks, these two citations here, a more comprehensive analysis further strengthened the validity of reported observations. Okay, so basically, what they do here raises questions about the effectiveness of existing message passing schemes and hypergraphs. A possible lens for such an investigation, which is this, where you want to investigate the architecture's ability to effectively encode higher order information, could be graph reasoning tasks suggested in this citation, Luo 2023. Additionally, the negative results observed regarding hypergraph level encodings paired with hypergraph level architectures warrant further exploration. Specifically understanding how to effectively augment hypergraph inputs with structural and positional information that can be leveraged by hypergraph level architectures is a promising direction for future study. So this, and then of course they talk about the different topological domains of interest, and then that's the paper. So that's interesting. Thank you for bringing that paper to my attention. Let's see if we have things in the chat here. So we have this link from MorganMelanieWeber.com, one of the authors of this paper. So this was the third author, Melanie Weber from Harvard. Let's see the publications. So this is pretty much this sort of topological graph neural network type stuff. There's some citations under her publications that might be of interest here. We also have this, this is her GitHub repository. This is actually hypergraph encodings. And so this is actually the repository for GOML, which was actually for this paper. So this is their repository. And this shows, okay, so this shows some simulations here. So these are the hypergraph encodings. And just showing, so this is, okay, so this is, I guess, a graph. This is the hypergraph embedding. And then this is a random walk around the hypergraph. So, yeah. And what they're doing in the graph is they're doing a random walk. You can also do a random walk on the hypergraph. And then you have this hypergraph random walk here. So thank you for those links in the paper again. So did we have any comments or questions about that?
Yeah, I just thought, yeah, it's from that same kind of community that does, what are they called, like symmetry and,
oh yeah,
I've got their thing here, symmetry and geometry and neural representations.
Yeah. Oh yeah, that's the group. Yeah.
You know, and it's this, yeah, just like her CV is you know, absolutely very typical for that community.
Yeah, yeah.
And yeah, but interested, well, like they say, you know, it's about, I mean, the improvement is seen in these message passing or transformer graph neural networks, and you know, and tasks that have been, what was the term they used? Recast? No.
Reparameterization or renormalization, I guess.
You know, as hypergraphs.
Yeah.
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 So just yeah.
Yeah, I think that's an important point, and we haven't talked about that in that context so far, because we've talked about, we've done kind of the operational stuff of creating hypergraphs and graph data. I mean, we have like the connectivity matrices, and then we have these hypergraphs that we've created, and of course, your graph nodes represent, say, cells, and then the hypergraphs represent categories. Sometimes they represent, you know, other things, and you can have things within cells. So each cell could also be a hypernode with different attributes in them. So like you could have, you know, different mechanical properties, different types of proteins represented, different types of gene expression represented. And then, so all of these things are kind of nested categories. So your hypernodes are basically some set of nodes and how they're structured. But, you know, you also have to think about this reparameterization or renormalization. Every time you move up a level, you have, or every time you're using some sort of, you know, set of relations. So say we were interested in, like, for every cell in the C. elegans, we're interested in the complement of gene expression. We have this for different genes. We've seen this before many times, where each cell will have, you know, you'll be interested in gene expression for different genes, maybe different, you know, different, like, I guess, complements of genes and their expression. And but the point is that those are different sets of relations. And so then that can be, you know, your cells can be hypernodes, your complement of genes can be hypernodes, and you can treat it like that. But then you have to also renormalize the graph, I guess, for each time you do that. And then you also ideally would have to also have a benchmark, from what I'm understanding. So they also talked about how you need to properly benchmark these against something else. And in our case, I don't know what we would benchmark it against. A lot of times people will, in network science, I know they'll use like random networks, or they might use some other type of network that's where, you know, the connectivity is known. You could perhaps create pseudo data to have something that kind of know what the distribution is, so you can compare it with what your result is for your experimental graph. So there are a lot of ways you can make comparisons, and I think it's just kind of speaking to some of these other ways forward. So instead of just saying we're going to create a hypergraph from our graph, we're creating a hypergraph. We have multiple sets of relations we can create the hypergraph from, and we need to renormalize that. And then we need to also compare it to something else to make sure it deviates random in the ways we expect. So, I mean, there are a lot of things to consider. And there is this community, the Symmetry Geometry Neural Representations group. They're they're often represented at NeurIPS. So they usually run a workshop there every year. And, there's also the Graph Neural Networks Conference, where people talk about symmetry, they talk about hypergraphs. I've seen people talk about hypergraphs at that conference. And message passing is a huge topic as well. So yeah, this is all very good stuff. I guess Soham and Mehul, if you have you have any comments or questions?
Yeah, so actually I missed a few little context in between. But I was thinking about benchmarking. So how could we do benchmark in our case? So whether it can be based on the techniques we are creating graphs or hypergraphs. We can't benchmark based on data, definitely. So what I can think of is different techniques or different distance thresholds, like we were discussing last week. Based on those things, we can do some kind of benchmarking.
Yeah, it's usually, it would be an idealized measure, I guess, would be a good way to do it. Because it's really hard to do a biological benchmark. You could do another species, you could create graphs of another species, but there are a lot of difficulties with making good inferences from that. You could have a lot of times in C. elegans research, and this is just kind of from experimental perspective, but they'll use a wild type and then have mutants. But of course the wild type is what we're kind of dealing with, so we don't have a good benchmark against a wild type just meaning a normal sort of C. elegans as opposed to a mutant, where they're things that deviate from the normal genotype. So we have, but I think a benchmark would be maybe something where, you have an idealized distance threshold. Maybe the distance thresholds are all uniform. Maybe they're kind of according to a normal distribution or something like that, and then you test what you have in your data set. And there's no biological relevance to maybe a benchmark, but it does give you something to base your comparison off of. If we have different samples, if we took a bunch of cell tracking data and we broke it down into 10 parts and we just used that as our sort of our test cases, how would that all compare to a benchmark? Or, how does, I don't know. I'd have to think about that more a bit.
Yeah, makes sense. Probably I will also try to think about it and find some relevant literature.
Okay. Yeah, that's good. Yeah, again, it's going to be a little tougher with biology because, I think a lot of the stuff in the graph neural networks literature is really kind of theoretical. It's a kind of theoretical machine learning. So people aren't actually throwing data sets at it per se. They're just working on the architectures, and they're working on, maybe at best they're working with very highly idealized data sets. They're not actually trying to get performance metrics and optimize them, or whatever they do in regular applied machine learning.
Yeah.
All right. So yeah, we keep losing Soham, but I wanted to ask Soham about this. But anyways, we can come back to this later if you want. I think that's a good thing to read for our people are interested in working in hypergraphs. it brings up a lot of interesting issues, and maybe we'll follow up on that next time. I'll go over it again and see if I can find something that's complementary to that, or if there's some other issues we want to raise and think about. That would be good as well. So thank you again to Morgan and Mehul and Soham for bringing that forward and discussing that. So I wanted to talk about this paper that came out. I think it was this last week. It's a really interesting paper. This is more along the lines of like biocomplexity. So I present you with this figure, and this is the paper. This is a paper, the title is Life Sets Off a Cascade of Machines. And machines being, I guess, biological processes. And so this figure is from the paper. And this graph, okay, so the x-axis is a log normalized size over meter, or size in meters, or size over meters. So it's basically the size of this phenomena, or the scale of phenomena. So you have, from left to right, you have atoms, macromolecules, self-reproducing machine, and that's in the realm of prokaryotes, as we'll get to in a minute. So we have spores and other cellular systems, we have cellular systems, protocells, things like that. And then we have development, and then we have evolution and ecology, which kind of extend out to the largest scale. So in blue, they kind of describe specific systems. At atoms, we have electrons, protons, photons. Then we have, moving up a little bit, small molecules and water. And then we have proteins, DNA, RNA, supramolecular complexes, viruses and organelles. Supramolecular complexes, viruses, and organelles are all these in this macromolecules category. Then we have membranes, so we're starting to move into cells, and then prokaryotes. So prokaryotes being a type of cell. In between membrane and prokaryotes, we could put these protocells. We could put maybe some other types of cells, so we have prokaryotes, we have eukaryotes. So we have viruses down below organelles. Organelles being like parts of a cell, a eukaryotic cell. We have membranes. Then we have prokaryotes. Then we have eukaryotes. So eukaryotes are more complex than prokaryotes. They're up on this other axis, which is log normalized time, and that's in seconds. So the time scale is compared to the size scale. And then from eukaryotes, we have multicellular, we have colonies. So multicellular and colonies are kind of, they're similar in a number of ways. When they say colonies, they mean like cells that live in colonies. Multicellular just means multicellular aggregates. And then we have populations. And that's usually some sort of organism, ecosystem, and biosphere. So, and then we have these two trends. So the first trend is this microcascade. And that's where time is equivalent to L to the fourth. So this is exponential. This is the early part of this curve. And then we have this sort of logarithmic phase, which is macrocascade, which is where time is approximate to L. So we have time approximate to L to the fourth, time approximate to L. And these are micro and macro cascades, respectively. So this is interesting that we get this phase transition here at this area of self-reproducing machine. So this is a very intriguing figure. And so let's get into these articles to tell us more. And this was something I think Morgan posted in one of the Slack channels. And I've seen this around too. This is from Derek's Mind blog. This is something to check out. This article here is Life Sets Off a Cascade of Machines. And so a fascinating PNAS article by Tlusty and Libchaber, which we'll get to in a minute, offers an oversimplified language of life. It is a long article, but I found it a very worthwhile read. So the significance of this article, this paper follows an idea by Leibniz that life can be seen as an infinite cascade of machine-making machines, down to atomic machines. It proposes an oversimplified language of life, highlighting certain scaling aspects and the key step of self-reproduction, with a singular point at 1 micron in 1,000 seconds. So what they're basically saying in this figure, and this is a blurred version of the figure, is that we start with atoms, and we can take this reductionist view, we can go all the way down to atoms. And these are components to these machines. And so atoms and macromolecules are all components to a machine. And when you get up to what they call the self-reproducing machines category, which is incidentally where this phase transition occurs, we get these reproducing machines or machine-making machines. And so this is, something where I think a lot of people have kind of noticed that there is this sort of hierarchy of systems in life. So life is organized, as they say, vertically, meaning that you have this vertical organization from individual units to these large-scale structures that are emergent phenomena. So, you can go to any biology textbook and see the sort of vertical organization of life. But then the question is where do you get self-reproduction? That is, where do you start to get things that sort of are self-perpetuating? And one might argue that you see that with like DNA and RNA. And in fact, in early life, you might see that as like the primary mode of self-reproduction. But what they're talking about here are these machines. So you have to have all sorts of different components to have this self-perpetuating machine that produces metabolism and reproduces itself in full and everything else. So you see these self-reproducing machines right around the time prokaryotes emerge. So that's where sort of they put this sort of phase transition between all these different processes that perpetuate these self-reproducing machines. So development perpetuates self-reproducing machines. Evolution and ecology also do this. Whereas macromolecules and atoms may be able to reproduce, but not in this sort of systemic sense. Okay, so here's the paper in PNAS. This is Tlusty and Libchaber. They talk about multi-scale organization, dense with interconnected cycles and loops, is one of living matter's most striking features. And this paper proposes a view that is also the most fundamental one. Concretely, we speculate that life can be presented as a hierarchical cascade of machines making machines. This simplified language of machines is false about the reality of living matter, but we hope it is still a useful vehicle to talk about the extreme varieties of life. So this is an important point. People like to use machine metaphors for biology, especially when you get down to the molecular scale. In the case of molecular machines, we do have molecular machines. You can think about it like a machine as being highly efficient and things like that. And life doesn't really have those same properties. But they're using this as sort of a vehicle to talk about, these processes, hierarchical cascade of processes making other processes. And that would be very similar. In our cybernetics group, we talked about closure of systems, so systemic closure. And this is, I guess, kind of coming from the same perspective where you have these self-perpetuating systems with these processes that can sort of reproduce themselves. So a lot of times you'll have processes that are sort of, not self-perpetuating. You put energy into a system and it runs until the energy runs out. In a self-perpetuating system, you get multiple systems that don't require inputs from the outside world, that you can have things that run kind of on their own. So like something like reproduction, or, if you have a seed, you can carry a seed around in your pocket, put it in the ground, and it'll sprout. And that genetic information is there, and as long as it doesn't get degraded by like UV radiation or by being dissolved in water, it can plant itself in different places and grow. So these are all things that are, kind of what they're getting at with this. When they say machines making machines, it's simply a metaphor. Thus, the paper is more about assenting certain scaling aspects of life, rather than about the infinite intricacy of life itself. And we hope there should be a mathematical language that captures life more fully than a purely realistic view. Such languages should fit their subject, living matter, like the formal language Lavoisier devised, fit his elementary theory of chemistry. And this is described as that the art of reasoning reduces to a well-formed language. So that's what they're getting at here. To begin this discussion, let us gaze for a moment at the rudimentary bacterium and imagine for now this minute creature that has two infinite cascades emerge, and that's just shown in figure 1. So they're talking about bacteria. They're talking about this microcascade that comes from so this is prokaryotes. There are two cascades that kind of emerge from bacteria, and this is from the perspective of bacteria. There's a microcascade which goes from the structure of the prokaryote cell down to its smallest parts, to atoms, and then we have this other cascade, this macro cascade that goes out towards development and evolution and ecology. So that's what they're kind of using prokaryotes as sort of the focal level of organization for self-reproducing machines. So, so the microcascade proceeds from the prokaryotic cell down to its tiniest parts, which are the membrane, the molecular machines inside the membrane, ribosomes, the cytoskeleton, the submachines that make proteins, RNA, and DNA, the parts that make the submachines, amino acids, nucleotides, and lipids, and all the way down to atoms, electrons, protons, and photons. And then, of course, the macro cascade in the other direction assembles bacteria into larger, more intricate organizational machines. So these are communities of bacteria, eukaryotic cells formed by the symbiosis of archaea and bacteria, tissues made of eukaryotic cells, organs constructed from tissues, organisms composed of organs, populations of organisms, ecosystems, and the biosphere. Okay. So in exploring this cascade, our starting point is the general notion of a machine and the specific questions of what makes living machines unique. The cascade of machines appears as a solution to the problem of survival through self-reproduction, realized in the world of salty water. We then ask how the machines self-organize into a cascade and elaborate on its scaling, especially on what determines its fundamental space and time scales. This is followed by a discussion of salty water, the world in which the cascade emerges and evolves. We examine what this physical boundary condition implies in realizing von Neumann's self-reproducing machine, particularly the role of electrodynamic forces. We conclude with tentative thoughts on the divergence of the macro cascade. So this is interesting. They kind of use von Neumann's self-reproducing machine as a sort of maybe a physical boundary condition for what self-reproduction entails. So if you're not familiar with von Neumann's self-reproducing machine, it's this cellular automata tool that von Neumann developed to look at different structures and how they can self-reproduce. What kinds of things need to be in place for self-reproduction? So this is, kind of getting into machines and their self-reproduction. So to follow Leibniz's inquiry into living machines, we must first explain what machine means. Broadly speaking, a machine is an object that transforms a physical system non-randomly to perform a definite function. Of these features of the machine, two require further clarification. So the first one is non-randomness. So you have to have this non-random component. It can't just be a random process. Machines might be very noisy, especially molecular ones. So machines that are living machines tend to be noisy, but they also tend to have a high rate of sort of fidelity. So like if you're looking at something like translation in the ribosome, that machine is very noisy, but it also has a pretty good level of fidelity, enough to produce amino acids and proteins that can, encode things that are needed for this larger machine, which is the cell. Yet, even the noisiest machine must have a non-random bias, or else it cannot function. So there has to be some sort of non-random bias, and indeed, in the example of the ribosomes, we see this non-random bias in the tRNA pools that take codons from the DNA or from the RNA and translate it into amino acids. So this is, again, we have this non-random bias. Molecular motors move erratically but in a definite average direction. Ion pumps transport charges intermittently but consistently across the same polarity of the membrane potential. So there's this sort of random walk going on, but the random walk is biased in a way that leads to this sort of outcome. And the division machinery splits cells and never merges them back. We see how machines inject local order into a universe drifting towards global disorder. And so this is where we have to talk about machines being dissipative in the thermodynamic sense. Second, we need to clarify the notion of functionality. Machines transform the system in a predictable manner, allowing them to perform tasks. By the scale of the task, the scale of the machine. So, for example, nanometric pumps convey ions, microtubules assemble into a micron-sized machine that organizes the chromosomes in the cell, and cells themselves form tissues, and this applies up to the scale of the biosphere. The existence of a function assumes no purpose or goal. These external human notions are beside the point. All we know is that the machine pushes the system along specific non-random paths in space-time, and these paths are functional. This is an interesting thing about purpose and goal, because there is this sort of debate historically about what they call teleonomic systems, where you actually, at some point, these kind of systems evolve some sort of goal-directedness. So in other words, you go from like a random walk to what we might call a ratchet, and there are like all sorts of molecular ratchets that you can observe. And that's what I was talking about back here, where you have processes that are noisy, but yet go in one direction. They allow for the sort of fidelity of a process that gives you something at a higher level. So like the example of the ribosomes or some other system, that they have this sort of this bias, and then you end up sometimes with goal-directedness. So in organisms, you have goal-directed behavior, where organisms are moving towards food sources or light sources. And so at different levels, you get this increasing goal-directedness from basically random processes. And the question is, how does that happen? And there's a literature on teleonomic systems, which are these programs that evolve in these kinds of systems that allow for goal-directedness. But that's kind of an aside from this article. I just wanted to mention that. Okay, so all we know is that this machine pushes the system along specific non-random paths in space-time, and that these paths are functional. This is where thinking about life in the language of machines, as Leibniz did, lead us away from the realm of pure physics and chemistry, where function is a meaningless word, and into the realm of engineering and technology, where function is everything. So I think there's this aspect of, like, when people think about machines and biology, that we have to understand that we're thinking about machines in an engineering sense. And there's certain baggage that comes along with an engineering approach to function. There's also this baggage of function as being this thing that's sort of, that we kind of ascribe characteristics to it. So when we use the term function, we kind of think of like maybe human behavior or something that is, everything has a role. And that's just not the way biology is. So we have to think about, like, when we say biological machine, how does that deviate from like engineering and technology versions of function? How does that deviate, say, from like human societal versions of function or human behavior versions of function. So it is a kind of a different, we can use the same language or that, but it's a different set of things that we're kind of interested in. So when molecules collide and exchange electrons and atoms, this chemical reaction serves no particular function by itself. Yet if this collision occurs on a living machine, say at the catalytic site of an enzyme, then it acquires a function through its context. So again, you have these random collisions that can occur in a chemical reaction. It doesn't really do anything. But if it occurs in a living machine, in a system where there are different types of, where it's heterogeneous, where there are different types of processes that are kind of interlinked in the same space, then it has some sort of function. And so maybe function isn't the right word. Maybe we need to rethink the language there. And maybe it might clarify things a bit. So what is the function of life? And they argue that the universal function of life is survival. So something has to survive to be functional, or something has to be relevant to survival to be functional. And that makes some sense, because if you think about the example we just gave, you have this process that's nominally random, but it works towards improving the sort of the survival or the fitness of, say, the cell. If you can trigger some sort of process, some sort of metabolic process, then you can survive. And a process that can be biased towards activating that metabolic process is probably better or more functional than one that doesn't. This global task unifies all living machines. Thus, we can propose looking at the living organism as a survival machine that is part of a well-coordinated double cascade. And this double cascade is from the figure. Survival is the physical notion of continuity in space and time. Systems with continuous space-time trajectories survive, and the living machine excels at selecting these continuous trajectories as compared to random physical processes. Other systems with different organizing principles might have emerged and disappeared, but we observe only the ones that persisted over time, those that survived. So what we see in biology, and especially with respect to these biological machines and biological function, is everything that survived. So we could have had cells of purely random processes. We could build, say, for example, a protocell with random processes in it. And that doesn't mean that the protocell can reproduce and sustain itself. It just means that you can put a bunch of chemicals in a bag and have a series of reactions that aren't linked and don't have this sort of autocatalytic potential, which means that is to say that one process initiates another process, so there's this sort of cycle that is tightly integrated and contributes to its reproduction and its survival. So we always have this observer bias, on what we see in nature. Only two extreme solutions of the problem of survival are known. The standard solution is physical robustness, as ancient rocks survive through our geological eons, or rivers flowing along slowly changing paths. The other extreme is the infinite cascade of life driven by self-replicating machines. So we have physical robustness, which is in physical systems and geomorphological systems. And the others are these cascades of life driven by self-replicating machines. For the living machine, survival is a subtler meaning with a different notion of continuity. Self-replicating machines must always remain continuously functional, implying that all levels of the cascade of machines must always be in synchrony. All fluxes of matter, energy, and information flowing through the levels in exchange with the environment must be balanced, requiring numerous feedback loops. Second, the machine must produce new copies of itself, and the lineage of machines ensures continuity over long time scales, not continuity of matter, but rather continuity of functional organization through the repeated cycle of self-reproduction. So this is where we have this aspect of continuity as well as function. So things must be continuously functional. You can't have a cascade of machines that are asynchronous or kind of trigger at different times. These processes all have to be synchronized and in this sort of cycle that where everything is tightly ordered in time. So you can't just have random fluxes, you have to have this order. And this is something that just, doesn't happen. It emerges over time and contributes to survival. And then, of course, you need to be able to reproduce new copies of yourself, and being able to do this over long time scales. So we see reproduction with DNA and RNA, but at the level of, say, like a cell, and they use eukaryotic cells as an example here. This is where you start to get this entire sort of set of reactions that are linked. So you not only get DNA and RNA that are reproduced, but the entire cell itself. A cell can divide and differentiate eventually. So it can reproduce itself in numerous ways. Okay. So then they talk about von Neumann's self-reproducing machine. And this kind of is a computational approach, which, of course, John von Neumann developed. The concept of the machine he envisioned was simple. It is a chimera of two conjoined submachines immersed in a sea of elementary building blocks. The first is a constructor that collects blocks from the sea and assembles them into an offspring machine, following the directions encoded along the blueprint tape. The second submachine is a copier. So you have a constructor and a copier. The copier replicates the tape and passes it on to the offspring machine, which is ready to produce the next generation. And there's a loop of doubling reiterates to infinity as long as it is fed with building blocks. So this is kind of like something that a very sort of computational approach to thinking about self-reproduction, but it's a way that you can think about it. So this is, on the left, you have von Neumann's architecture for a self-reproducing machine. You have a constructor and a copier in the machine, and then you have this tape, and you can think of this as maybe like RNA, where you can, make copies of the RNA or make from an RNA to a protein through this constructor and copier. So you're copying information, then you're constructing a new sequence of information. So you have the parent, which copies the machine, the offspring, which is where the constructor reproduces the machine. So it's basically copying and constructing. And those are two different processes. They're not the same process. You have to be able to copy, or faithfully copy something. You don't want too many mutations. And then you have to construct it. So you have to know the order, and you have to know sort of the geometry of the offspring. And so this produces, some sort of reliable, reproducible next generation. And this continues along the second trajectory that we talked about, the macro trajectory. And then this, of course, is an example of a self-reproducing machine in a cell where we have a membrane with DNA in the cells and then RNAP. And then this is where we have cell division. So we can see this parent to offspring process of the self-reproducing machine, where we have a constructor and a copier, and the membrane is sort of, creating two cells. All of the contents of the cell are segregating to either side of the cell, of the original cell, and the membrane is enclosing these into two separate cells at the end of this process. So we can map a von Neumann self-reproducing machine to a bacterial cell division, a cell doubling event. Okay. So this just kind of goes over more things about complexity and self-reproduction, talking about von Neumann's machine and kind of contemplating the role of this sort of reproduction and in biology, in biological machines. And then talking about loops and cycles and feedbacks, which are, of course, important for linking together these processes, because a process, of course, in isolation can have internal feedback, but when coupled to other processes can form these self-sustaining reactions and these self-sustaining sets of machines that form, say, like a cell. And so you get these cascades of machines, and then you get these doubling cascades, and you get further complexity from that. Okay, so that's, I don't think there's anything else in the paper I wanted to cover. It just really gets into more things about like information theory and protein machines, and then cells, life's quanta, which is talking about the cell as the fundamental unit of biology. And then, of course, this figure here, which is sort of the criticality of this. So we have this example from sort of biology where we have the size L, which is in microns, and the doubling time, which is in, I guess, seconds. And then we have the membrane cost here. The low surface volume reduces flux mass here. So these different aspects of the cellular morphology or physiology are a part of this curve. So you have the membrane cost here, where surface volume reduces flux mass. And there's an optimal point in this curve, which is where you find ribosome self-reproduction time. So the doubling time is 10 to the third. The size in microns is 10 to the 0. So this is like a theoretical curve that describes a lot of things going on in the cell. So doubling time is a function of cell size. At the critical size, L sub c is around 1 micron. The microbial cell achieves the fastest doubling time, which is the lower bound set by the self-reproduction time of the ribosome. So the microbial cell is sort of this lower bound of what, I guess theoretically the lower bound of what you can have for these self-reproducing machines. Larger cells, where L is larger than L sub c, has a lower surface volume to ratio and therefore self-reproduce more slowly. In smaller cells, where L sub c is larger than L, the cost of producing the membrane of width around 10 microns, or a changing width of around 10 microns, or actually nanometers, slows down the doubling as this relation holds. So this is kind of thinking about this. This is L sub c at this optimal point. This is T sub c here for the doubling time. So the size and the doubling time are optimized in the prokaryotic cell. And then they talk about feeding the cascade. So this kind of talks about some of these processes and the optimal energy flux needed. And then finally talking about salty water and sort of where the boundaries of this cascade are. So if you think about a prokaryotic cell in salty water, that's the sort of the boundary condition for these linked cascades. So at the beginning of life, we had these cascades. We had RNA world. We had these cascades that were enclosed by a membrane. The membrane enclosed these processes, and salty water was outside. And so this is something that provided energy and nutrition to this emerging cell. So they talk about that here. So that's, I think, all for that paper. That's something that I wanted to go over. I thought it was a fascinating paper, and it has a lot of relevance to early life. So we've been, doing some early life research in the group. It also has relevance to biophysics and biocomplexity. And it really, helps you think, I think, about this idea of systemic closure and how you go from processes that are sort of out in, that are sort of independent to processes that are linked in a living system and that are self-sustaining and self-reproducing. And then thinking about the computational analogy to that, which is the von Neumann self-replicating machine, which is in the cellular automata space, but then also describes this process and abstracts this process and serves as sort of almost like an embedding for early life. Okay, do we have any comments or questions about that?
Really interesting. I haven't, I'm on my school run here, so I can't do it justice, but yeah, was really interested to dig into that, and seemed like a really interesting paper.
Yeah.
Oh, I put a comment in the chat.
Okay. So Susan says, Dick found an interesting paper. Wnt signaling modulates mechanotransduction in the epidermis to drive hair follicle regeneration. Okay, that's something we should check out. Okay.
Well, it fits in with what I was doing with epidermis.
Oh, right. Yeah.
And the elasticity. So apparently hair grows at 5 to 15 kilopascals.
Yeah, it looks interesting. So wind signaling modulates that process or the elasticity, or
Yeah, it's hard to say. It's one of those chicken and egg things. Is it wind that's causing the adherence junctions to form? So then there's more pull on the cell, like it cramps, or is it these differentiation waves? Like is it cell cramps or I don't know, the adherence junctions connected to the cell's actomyosin ring on the top, because this happens in epithelial cells. So this is an epithelial cell thing. And so is that what causes Wnt to be produced? Or if you have Wnt, then you get more adherence junctions, and therefore you get more cramping. I don't know, it's all like the chicken and egg thing. Like it's a kind of a circular process.
yeah.
Yeah, and I guess Vic's comment here is it says wind signaling mechanoregulatory role that manipulates the machinery of mechanotransduction to drive regeneration, or vice versa. So it might be like they're saying Wnt is the key, but it might be that the mechanotransduction produces Wnt, which produces more connections. And that's true if you have more connections. If you have more force, you get more connections in a cell. It automatically produces them. I don't know. I was just sort of being a biologist and reading about observation and sticking them in my paper and saying, see, this happens. But,
Yeah.
yeah, it says under intense mechanical load, such as those seen during bone formation and tumorigenesis, beta-catenin translocates to the nucleus in a force-dependent manner. So it's changing the production of something using our DNA. And yeah, that also could be during a cramp, like if the cells cramp or the tissue that undergoes a cramp, then that's an intense mechanical load automatically.
Yeah.
So that fits with that. So that's why we're saying differentiation waves, because they're cell cramps.
Right.
Anyway, it's an interesting paper.
Yeah, it sounds like it. Maybe we'll come back to that next week. But thanks for going over the
that one might be worth money if you can figure it out, because everybody wants to grow hair over there and not over here.
Right.
Everybody's fussy about their hair, and it's just
So you just like crimp your skin with clampers, and you just keep crimping your skin, and you'll get hair. Yeah.
My suggestion is add more elastin to your aging scalp.
Yeah. Oh, yeah.
Mine too. Like
just rub it in. Yeah.
Anyways, that's I guess what I've been looking at it as an aside here. I'm supposed to be writing a paper.
Yeah. All right. Well, that sounds great. Thank you, everyone, for attending. if that's all for today, see you next week.
Okay, bye.
Thanks, bye.